So the question was, does the internal pH of the cell affect the cancer? It doesn't seem as if the internal pH is the important thing. But the immediate surrounding pH is. So what happens is that the pH around the cancer cell is acid, and that seems to have an impact on signalling, including this hypoxic signalling process. Yeah, but they might be a little bit acid, but they maintain their internal pH quite tightly, like all our cells do. There was a question further back. Yes, you have to shout.
Mine's slightly different. I'm looking at my own situation in terms of diet.
Last September, and I'm now at a stage where I'm having my 3 monthly scans. I've been told by the Marsden that will last for 2 years. If everything's well, that'll then go to 6 months, possibly a year. My concern is I've still got nodules that at this time are not developing. The Glivik is holding them off, if you like. But my concern is if I go to 6 months or a year scans and something's developing within that space, where do I stand? Do I go back to 3 monthly scans, or is it just based on individuals' circumstances?
That's a very good question. I think the optimal time, the optimal time interval for follow-up is a real thorny question, because we try, and we're constantly being told by our radiologists to try and restrict exposure to radiation. And yet we know that superficial peritoneal nodules are not very well seen by MRI or ultrasound. Ultrasound is great for the liver, pelvis. MRI is pretty good, but doesn't perhaps pick up small peritoneal nodules as well as CT. So the question is, I suppose, a trade-off between risk and benefit, and every patient, all I can reassure you to say is that every patient's considered individually, rather than being put into some sort of strict formula. And obviously, if there are anything that's actually suspicious of active disease, then you wouldn't extend the interval between scans.
What relevance does mitotic count have on the decision to give adjuvant therapy, and for how long? In my case, my mitotic count is 62 over 50.
That's quite high. Well, in my view, mitotic count is probably the most important factor of the various of size, site, and mitotic count. Mitotic count, I think, trumps the others in terms of risk. I think that would be clearly in a high-risk group with that mitotic count, whatever the tumour size and whatever the site, and hence the argument could be made for giving adjuvant treatment. Now, I know that NICE said no on the grounds of cost-benefit. They thoughtfully managed to announce that they would defer the reconsideration of adjuvant therapy on the very day that the Scandinavian data were being presented in Chicago, by some strange coincidence. Very little we can do other than use the other avenues open to us, which is go through a PCT, be told no, go to a cancer drugs fund. That's okay if you're in certain parts of the country, no good if you're in London yet. We're trying to get approval for the London Cancer Drugs Fund, but no guarantee we will. So it's an ongoing battle. But I think, yes, mitotic count, very important, and can certainly be a key part of the decision to give adjuvant treatment.
I'd just like to ask about the mitochondrial organelles. You were saying that the cancer cells have very few of the mitochondria to produce the energy they're producing their own. The mitochondria that are actually there, does the mitochondrial DNA still exhibit the same as that within the mother?
Yes. Mitochondrial DNA, yes, it's maternally transmitted, and there are conditions where mitochondrial DNA is actually responsible for an inherited disease, muscular dystrophies, if I recall. In other respects, we know very little about it, at least I do. I'm not a genetics expert by any means. To what extent mitochondrial DNA might be involved in malignancy, I honestly don't know. We do know there are other things called microRNAs which control gene expression, which are being actively explored in a whole range of cancers at the moment, about which we knew nothing much 10 years ago. So there's lots of work going on, but I don't, I'm afraid, know much about mitochondrial DNA. Question now. Sorry, I think better. Excuse me. Hello. Nice to see you.
Have you got any more views on doubling the dose of GIST?
Dub-
not GIST, obviously.
okay.
And also, is there any trial in the UK? You mentioned quite a lot of new drugs. I'm interested. Thank you.
There aren't any. No, there aren't any studies looking at dose escalation. I'm going to look at Roger here. You get me on one of my hobby horses here. Roger and I took this issue to NICE, along with the help of many others, and unfortunately the repeat appraisal of imatinib, which was promised for 2007, didn't happen till 2010, and it was then rigged so that the benefit of the higher dose in exon 9 mutant GIST was not even considered. They didn't permit those data to be discussed. They restricted the appraisal to patients progressing on 400, so that they could do a multi-technology appraisal head-to-head against sunitinib. Result predictable, imatinib more expensive, not permitted. So they didn't even look at it. Novartis, unfortunately, never registered the higher dose. So we were on hiding to nothing from the start, because, strictly speaking, NICE could then hide behind their rule which says we only do technology appraisals on licensed medicines. So 800 milligram, not a licensed medicine, therefore we're not going to consider it from the word go. And we went round and round in circles and did all sorts of appeals, and to no avail. But because Novartis has no interest in a drug market as small as the UK, when the rest of the world are perfectly willing to give a bigger dose when they need to, I'm afraid we're stuck.
I was wondering if there's any likelihood of a marker in the blood to show activity of the disease.
That's a very good question. There isn't at the moment, but there might be. So what we're doing at the moment, at least what we've just started to do, is to see whether the mutation can be detected in the bloodstream, because there is naked DNA in the bloodstream from cells that have broken down. And in some cases you can actually detect a cancer-specific mutation in the bloodstream. And we have started to do this, and we have, on at least one occasion, found the very same mutation that we know to be present in the patient's tumour in that patient's blood. Now, of course, what you'd then have to say is, well, does it go away in response to treatment? Does it come back when the treatment is failing. The other thing which we're interested in is angiogenesis, and whether or not an angiogenesis inhibitor, the other way around, whether when patient's disease is progressing, whether proteins that are linked to angiogenesis like VEGF go up in a predictable way, and could that be a marker of early relapse? So there are some things that we're toying with, but there aren't at the moment any good markers.
You talked a lot about imatinib, but less about sunitinib. But do you have any more data about sunitinib, about its usefulness, be it progression-free survival and all that kind of thing?
Well, there are more data on sunitinib from sort of expanded access studies. Prior to license, or prior to the drug being reimbursable in all countries, there was a scheme for giving patients access to the drug, and the data have been collected in the same way as they would have been collected for a trial, and essentially confirm exactly what the randomised trial showed, in the sense that the percentage of patients benefiting is predictable. Duration of benefit again is fairly predictable. What we've learnt about toxicity is that alternative schedules such as 37.5 milligrams daily are more acceptable to most patients, seem to be equally efficacious, and really the message from talking to lots of doctors and lots of patients is experiment, and be prepared, to work with the patient to say, well, okay, so if your sore mouth comes on at 3 weeks, then maybe 3 weeks on, 1 week off might be the best schedule for you. Some patients say, well, I get very tired, but I'm better after 3 days off treatment. Okay, 3 days off treatment. There's nothing magic about a week or 2 weeks. A lot of patients will say that their disease-related symptoms actually come back during the time off treatment. So breaks are generally, we feel, not a great idea. And that 4 weeks on, 2 weeks off schedule was developed arbitrarily because fatigue was dose limiting, and they wanted to give the biggest daily dose they could get away with. That was from the phase 1 trial done in France at Gustave Roussy. So I think that, I had a patient who for a long time was stable on 25 milligrams alternating with 37.5 milligrams, 3 weeks on, 1 week off. That was the right regimen for her. So I think that we need to be more creative. We don't at the moment, I'm afraid, have a new drug specifically for GIST. However, in the drug development unit, we do have clinical trials which are highly relevant to GIST, looking at signalling proteins on the protein kinase, the PI3K, phosphoinositide 3 kinase pathway. So inhibitors of PI3K, AKT, mTOR, those proteins seem to be crucially important. There's a review by Mike Heinrich, which is going to be published shortly, which focuses on laboratory research saying the same thing, that PI3K is crucially important. So we have seen responses in patients with resistant GIST on a PI3 kinase inhibitor. And the likely future is combinations of drugs like that. We can't be specific at this stage, but certainly there is research on alternative targets, yes.